Okay, hello, assalamu alaikum.
Walaikum assalam wa rahmatullah.
So now unmute, now mute your mics so that you can have better listening from my side. So you, I believe you can see my screen now.
Yes, sir.
Okay, so we are going to start with the continue with the same topic that we finished last time. We were discussing the plant growth promoting rhizobacteria, and I briefly gave you the outline of the plant growth promoting rhizobacteria, what these are, and generally their mechanisms. So today we are going to discuss the detail of those mechanisms, how the plant growth promoting rhizobacteria, which I told you last time that these are getting more and more popular. So I gave you the list last time of these bacteria. So today I will discuss those mechanisms one by one. First of all, it is the phytostimulation. It is clear from the name even, phytostimulations means they stimulate the plant growth. So they have direct growth promotion mechanism by the production of phytohormones, and those phytohormones include indolestic acid, abscisic acid, cytokinins, gibberellins, and ethylene. These phytohormones, if you remember, I told you last time, the difference between the phytohormones and the fertilizers is that the phytohormones, they affect the physiological processes at even very low concentration. So the phytohormones enhance the growth by virtue of their effect on what they do. They affect the cell division, cell enlargement, seed germination, and root formation, as well as the stem elongation. So these are the mechanisms how the plant growth promoting rhizobacteria, they produce these phytohormones, or you can say the plant growth regulators. And these phytohormones, they affect the plant growth through these mechanisms, through cell division, cell enlargement, seed germination, like that, right? So the second mechanism is biofertilization. So first one was the phytostimulation. Second is the biofertilization. So biofertilization means that the PGPR, they increase nutrient availability by increasing nutrient concentration in the rhizosphere. You know that the PGPR are present in the rhizosphere. So due to one or the other mechanism, they increase the nutrient concentration in the rhizosphere. So as a result, more nutrients are available to the plants, so they can have better growth when they have better nutrient concentration. For example, the phosphate solubilizing bacteria, they solubilize phosphate by various mechanisms like production of organic and inorganic acids, release hydrogen ions, production of chelating substances, and through enzymes like phosphatases. So this is how the PGPR, they increase nutrient concentration in the rhizosphere. So as I said, they can improve the solubilize the inorganic phosphorus which is present in the rhizosphere. And this is done through the production of certain organic and inorganic acids, or the release of H ions.
Excuse me, sir.
Please,
Excuse me, sir.
please mute
सर
your
कुछ
mic.
बच्चे भी वेटिंग में है सर, उनको एडमिट कर लें.
Mute your mics, please. production of chelating substances, and through enzymes like phosphatase. Okay. Then another example is Pseudomonas species. They stimulate calcium uptake. Silicate mineral solubilizing bacteria, such bacteria which solubilize the silicate minerals. You know, the silicate minerals have potassium in them. So these bacteria, and their example is Bacillus, so they improve the potassium availability in the rhizosphere. And the Pseudomonas fluorescens, it increases iron availability by the production of low molecular weight cytophores. I have many times explained that how it works. So it produces low molecular weight cytophores, which improve the iron availability. Then there are rhizobia, which can fix atmospheric nitrogen and make it available to the plants. So these are the mechanisms how the microorganisms, they improve or they increase the nutrient concentration in the rhizosphere, and due to that increase of the nutrient concentration, more nutrients are available to the plants. So as a result, they have more more plant growth. Burkholderia species, they can enhance the zinc uptake. Then the third mechanism is root colonization. When we say colonization means to reside on the roots. So root colonization, if the PGPR have to do something to the plant, they have to colonize on the roots. Only those bacteria can be effective which can colonize the roots. So the success of bacteria to enhance plant growth depends on its potential to colonize the plant roots. Those who can better colonize the plant roots, they can improve the plant growth better. And those who cannot colonize the plant roots, they cannot affect the plant growth to that extent. One of the aspects of better root colonization is the ability of bacteria to compete with the indigenous microbial population. You know, if the bacteria, they are unable to colonize the roots, then what can happen is that those bacteria which are already present in the soil and they are not useful, they will reside on the plant roots, and the beneficial bacteria will be unable to colonize the roots. So we inoculate or we add beneficial bacteria for the purpose of root colonization. So if those bacteria which we have added, if they are unable to colonize the roots due to competition with the indigenous population, then there will be less or no effect of the added bacteria on the plant growth. Most abundant microorganisms, better competitiveness for root colonization. So it's a very common sense that those bacteria which are abundant in the rhizosphere, which are more in population, they will have better chances of colonizing the roots compared with those which are in less population. The microbes use different strategies to colonize the roots and for their survival in this stressful environment. So how the microorganisms, they reside on the roots, how they colonize the roots, and how they survive in this stressful environment. So these mechanisms or these strategies include ACC deaminase activity. I told you last time ACC stands for 1-aminocyclopropane 1-carboxylic acid. Today I will tell you its detail also. So the ACC deaminase activity helps the plant to survive in the microorganisms to survive in this stressful environment. Then they produce exopolysaccharide, which help them in better survival and better colonization on the plant roots. They produce certain organic solutes, which help the plant to have more microorganisms in their rhizosphere or on their roots due to even under stressful environment. Production of antioxidant enzymes. If you remember last time I told you that there may be certain uh, reactive oxygen species in the plants which may be toxic for the plants. So these reactive oxygen species can be suppressed by the production of antioxidant enzymes. I will tell you what are these antioxidant enzymes. These help the plant avoiding the effect of reactive oxygen species. PGPR can produce different enzymes for their survival in biotic and abiotic stresses. So when we say biotic stresses, it means plant is the stress due to any living organisms. That may be a disease, and abiotic stress means due to some abiotic factor like temperature, heat, salinity, water stress, all these are the abiotic stresses. So when there is any biotic or abiotic stress, the PGPR, what they do is they produce certain enzymes. Those enzymes are cellulase, ACC deaminase, chitinase, and these enzymes help the plant to grow even under stressful conditions. The next mechanism is enzymatic activities. The microorganisms, they produce certain enzymes, and these enzymes, the reduction of elevated level of ethylene under stress by ACC deaminase activity. So if you remember last time I explained that under stressful conditions, plants produce more ethylene, which suppresses the plant growth. So under stress condition, more ACC is produced, and there are certain bacteria which produce ACC deaminase activity, and that does not allow ACC to be converted into ethylene. So as a result, the inhibitory effect of ethylene is suppressed. Similarly, some bacteria, they produce chitinase enzyme, which suppresses the diseases. How it suppresses the diseases? Chitinase, you know, the fungal cell wall is made up of chitin. And generally the fungi, many of the fungi are pathogens. So when the bacteria, they produce chitinase enzyme, these chitinase degrade the cell wall of fungi. So as a result, the fungi is suppressed, so the disease is suppressed. The enhancement of nodule formation by rhizobia due to the production of cellulase, which assist in bacterial penetration to root hair. The cell wall of the root hair is made up of cellulose. So some bacteria, particularly these rhizobia, they produce cellulase enzyme. And what these cellulase enzyme do is they puncture the root, and from that puncture, from that breakdown, the rhizobia, they can enter or penetrate into the root hair where they multiply, develop the nodules, and fix nitrogen over there. So these cellulase enzymes help the rhizobia to penetrate into the plant. Then another mechanism is growth enhancement through vitamins. So certain bacterial species, these produce certain vitamins that cause positive effect on plant growth and development. Vitamins, you know, even if the human beings, they take vitamins, that they can have beneficial effects. So similarly, the plant growth is also improved if they get the vitamins. And certain bacterial species or the PGPR, they can produce these vitamins. These vitamin producing Pseudomonas fluorescens has more root colonization ability. So such Pseudomonas species which have the ability to produce vitamins, they have better ability to colonize the root. Then the other mechanism is biocontrol activity. Biocontrol means to control certain diseases with the help of living organisms. So PGPR, or the plant growth promoting rhizobacteria, they act as effective biocontrol agents by suppressing the effect of diseases. So they can suppress the diseases. So when there is less disease, it means there will be more plant growth. What are the mechanisms of biocontrol activity? So these include competition. So when there are PGPR, they will compete with the pathogens present over there, and due to that competition, if the PGPR they win that competition, they will suppress the pathogens. So as a result, or all the resources which are available over there, the pathogens they have to compete with the PGPR for getting those resources. So PGPR do not allow them to have those resources freely available. So as a result, there is limited resources available for the pathogens. So as a result, the disease is controlled. Another mechanism is the antibiotic production, that the PGPR produce certain antibiotics which suppress the pathogens, certain bacteria which are the pathogens, they suppress those bacteria by producing antibiotics. डिग्रेडेशन ऑफ फंगल सेल वॉल, मैंने अभी थोड़ी देर पहले आपको बताया है कि I just told you that some of the bacteria they produce chitinase enzyme, and the fungal cell wall is made up of chitin. So the bacteria which have the ability to produce chitinase enzyme, it degrades the cell wall of fungi which is acting as pathogen. So as a result, the pathogenic effect of fungi is reduced. Then sequestering iron by the production of cytophores. We have already discussed this, that the PGPR, they make chelates with the iron which is present over there, and as a result, that iron is no more available for the pathogen. So due to the deficiency of iron, pathogens are killed over there. So as a result, the control of disease which is caused by those pathogens. Then chitinase, cellulase, and lyases, these are the well-known fungal cell wall degrading enzyme. So I gave you the example of chitinase, but there are certain other enzymes like cellulase and lyases which can also degrade the cell wall of the fungi, and as a result, the the diseases caused by those fungi or those pathogens are suppressed. The presence of Pseudomonas species which have chitinase enzyme inhibits the growth of Rhizocotonia solani by degrading its cell wall. So Rhizocotonia solani is a pathogen. If you have in the rhizosphere Pseudomonas species which can produce chitinase, these chitinase will degrade the cell wall of Rhizocotonia solani. So as a result of that, the harmful effect or the disease caused by Rhizocotonia solani is suppressed. Black rot of tobacco, which is also a disease, this can be suppressed by such Pseudomonas species which produce hydrogen cyanide. Pythium species can produce hydrophores, which can decrease the availability of iron for fungal growth. The bacterial hydrophores are involved in inducing systemic resistance that enhancing plants' defensive capacity against pathogen. This I have already explained that. So the PGPR can also produce antibiotics. We are discussing the mechanisms, how the PGPR can play a role in biological control against biotic or abiotic stresses. So PGPR also produce antibiotics. These antibiotics suppress the pathogen and also induce systemic resistance in the plant. Bacillus thuringiensis produce insecticidal protein. You know the BT cotton. What happens in the BT cotton is actually that the gene which are present in Bacillus species, Bacillus thuringiensis, such genes are taken from those bacteria and those are inserted into the plants, and the plants which have this BT gene, they have the ability that the sucking insects, when they suck the sap of that plant, those are killed because it is producing a sort of poisonous protein which has insecticidal ability. Then another mechanism is removal or detoxification of organic and inorganic pollutants. So
Excuse me, sir.
bacteria use different intra and extra mechanisms to detoxify the adverse effects of heavy metals in their tissues. So sometimes what happens is that there is the presence of certain heavy metals or certain other toxic substances, and the bacteria can take up those toxic metals. So what the bacteria can do, they can use those heavy metals, can detoxify those heavy metals, or can exclude those heavy metals from their body. These mechanisms include how they do this. This is through the production of proteins which absorbs heavy metals. They produce such protein that absorbs the heavy metals, or detoxification by taking them in vacuoles. They take them in vacuoles where it is detoxified. It is no more toxic for the plant. The mechanisms may vary among bacterial species and also for different metals. So different bacterial species have different mechanism against different metals, right? When you will go to the higher classes, then you will study in detail what mechanism is effective against what metal. But at the moment, you need to know that different bacteria have different mechanisms against different metals, how they avoid from the heavy metals, the effects of heavy metals. Microbes can detoxify zinc by binding it in the outer membrane by producing zinc binding protein. So the microbes produce zinc binding protein that binds with the zinc in the outer membrane and does not allow to enter into the cell. So those bacteria can survive from that zinc. Bacterial inoculation increases degradation of chlorobenzoates and pesticides in highly contaminated soils. So if there are highly contaminated soils and those contain chlorobenzoates or other pesticides, those can be taken up by the plants and can enter into the food chain. But if you inoculate with certain bacteria, suitable bacteria, those bacteria can degrade those toxic substances like chlorobenzoates or pesticides. And as a result, those toxic substances will not be taken up by the plant, so these will not enter into the food chain. The inoculation of Lupinus luteus with genetically engineered nickel-resistant Bacillus cepacia results in high nickel uptake by plant. So you have a soil which is rich in nickel, and you want to remediate that soil. What you can do is you can grow Lupinus luteus over there, and what you can do is you can inoculate it with genetically engineered nickel resistant bacillus species. Such bacillus species where you have introduced the gene of nickel resistance. So when those bacteria are inoculated to the lupinus, what happens is that then this lupinus luteus can take up more nickel from the soil. So then you cut that plant and the nickel is removed from the soil. So this is a sort of phytoremediation. Another mechanism is enhancement of photosynthetic activity. So it's clear from that it increases the PGPR, they increase the photosynthetic activity. Photosynthesis, you know, is the most effective or the most important biochemical process which is taking place on our planet Earth. So under stressful environment, there is reduction in photosynthesis, which might be due to decrease in leaf expansion. Leaves do not expand properly. When they do not expand properly, less light absorption. So when less light absorption, so there will be less photosynthesis. Premature leaf senescence, impaired photosynthetic machinery like photosystem 1, photosystem 2, in the whole system that can be destroyed or that can be, you can say, affected badly due to the certain reasons, those due to maybe stressful environment. Associated reduction in food production, there is less food production, so less bacterial growth or the plant growth. So how PGPR can enhance photosynthesis? They can enhance photosynthesis by enhancement in chlorophyll content. So they enhance the chlorophyll content. So as a result of that, you know, chlorophyll content is directly related with the photosynthesis. More the chlorophyll content, more will be the photosynthesis. They increase shoot and root length. When more shoot and root length, more photosynthesis. Increase in leaf surface area. More the leaf surface area, more the photosynthesis. Improving nutrient availability. If more nutrients are available to the plant growth, they will be available to have better metabolic functions. And the next mechanism is induction of stress tolerance, that the PGPR present in the rhizosphere, they can induce stress tolerance.
सर कुछ लोग वेटिंग पे है उन्हें एडमिट कर लें.
Okay, I have admitted them, but last time I told that after 15 minutes I will not allow anybody to enter.
सर वो नेट का प्रॉब्लम था उनका, इस वजह से
Okay, I have admitted them, but from now onwards I will not admit anyone else. Okay, so the induction of stress tolerance, that is another mechanism. So PGP are able to tolerate stress due to sophisticated signaling system. They have very sophisticated signaling system through which they provide signal to the other PGPR as well as to the plant that this particular stress is there. So you have to adopt certain mechanism to compete with that stress or to count for that stress. Then their exceptional genetic makeup. They have some special genetic makeup which allows those bacteria to grow even under stressful conditions. We will study an independent chapter where we will study how the bacteria, they survive under stressful conditions. What are those mechanisms? Then the PGPRs have protective measures. What protective measures these PGPR have? Number one is the production of exopolysaccharides. They produce exopolysaccharides and the accumulation of polyhydroxybutyrate, proline, and ectoine. All these are the different compounds which help particularly in tolerating the salt stress. They act as a sort of osmolytes. They do not allow the plant internal water potential to decrease, as a result of that, the plant can grow better. And as far as the bacteria themselves are concerned, if they produce, though if this is one of the mechanism how they survive under stressful conditions, that they produce these osmolytes in their body, and as a result, they can themselves survive. So these are the mechanisms how the PGPR, they can improve the plant growth. Now a little bit applied topic, how we can apply these PGPR for plant growth promotion. So we have learned by now what are the PGPR and how they can affect the plant growth. Now, how to apply them for improving plant growth, this is what we are going to study now. The application of PGPR for plant growth promotion. So there may be two possibilities. One is the growth promotion under normal conditions. Under normal conditions, PGPR act as efficient biofertilizers that promote plant growth through, थोड़ी देर पहले we did discuss the biofertilizers, how they play their role in improving the nutrient availability to the plants. So PGPR act as efficient biofertilizers that can promote plant growth through what are the mechanisms we discussed, through maybe through phytohormone production, through phosphate solubilization, through cytophore production, through regulation of hormonal level. So looking at this slide, you can differentiate between the effect of chemical fertilizers and the biofertilizers. When you apply some chemical fertilizer, it provides only the nutrients. But when you apply certain biofertilizer, it has some additional benefits as well. It can produce phytohormones, it can solubilize some nutrients which are already present, it can regulate the hormone level, and it can produce the cytophores. So these are the additional benefits instead of the chemical fertilizers, right? So that's why generally we say that it is always better to supplement your chemical fertilizers with biofertilizers so that you can have these additional benefits. But of course, these biofertilizers cannot fulfill the complete nutrient requirement of the plant. So we must add chemical fertilizer, but we should supplement that chemical fertilizer with biofertilizer. Only biofertilizers cannot do the job of chemical fertilizers. We have to add both to have additional benefits. Because our population is increasing at alarming rate, so we have to increase every grain in every crop so that we can successfully feed our increasing population. So that's why we say that we must supplement the chemical fertilizers with biofertilizers. But I did tell it in some earlier lecture as well, that if somebody claims that only the biofertilizers can completely replace the chemical fertilizers, it is not wise idea. It's not good idea. The chemical fertilizers are unavoidable. We have to add chemical fertilizers, but we can reduce the chemical fertilizers by supplementing it with biofertilizers. You can do this, that instead of adding 3 bags of urea, you add 2 bags of urea and add some biofertilizer as well, right? So you can reduce the chemical fertilizer, but you cannot avoid the chemical fertilizers. Inoculation with PGPR increases plant growth and nutrient status of plant, particularly nitrogen and phosphorus. The production of volatile compounds by the bacteria is another growth promoting mechanism. They produce certain volatile compounds which promote the plant growth. This was the growth promotion under normal conditions. Then the second possibility is growth promotion under stressed conditions. And I have already explained that the stresses may be of two types. Those may be biotic stresses or maybe abiotic stresses. So environmental stresses are the most limiting factors for crop productivity. Can anybody tell me some environmental stresses? Salman Niaz. Salman Niaz. What are the environmental stresses? Hassan Sajjad.
Yes, sir.
What are the environmental stresses?
Temperature, rainfall, climate change, sir.
Yes. So all these are the environmental stresses: salinity, temperature, moisture. These are the environmental stresses, right? So and these are the abiotic stresses. These are not biotic, these are abiotic stresses. So they can affect the crop productivity. So the abiotic stresses include salinity, drought, extreme temperature that may be too high or too low, chilling, of course, too low temperature, heavy metals. These are the abiotic stresses. And biotic stresses include insects, pathogen attack. So these biotic and abiotic stresses, they can affect the plant growth, reduce the plant growth. So first we discussed how the PGPR, they can be helpful in improving plant growth under normal conditions. And now we are discussing how the PGPR can improve plant growth under adverse conditions. So what may be the adverse conditions? We discussed that these may be biotic stresses or abiotic stresses. So PGPR use various mechanisms to combat the stresses like salinity, drought, heavy metals, nutrient stress, and pathogens. So how they do that? Before that, just try to understand this figure which I have given here. So what I have said is that these may be the stresses present in the soil environment. There may be some pathogen present in the soil environment. There may be flooding. There may be salinity, drought, temperature, or heavy metals. So these may be the stresses. And these stresses may have negative impact on plant growth. So you can see the negative impact on plant growth and how that negative impact takes place. It is in the form of leaf abscission. It may be in the form of leaf abscission, senescence, nutritional imbalance, hormonal imbalance, root inhibition, or shoot inhibition. So due to any of these reasons, the growth is inhibited. But when you inoculate with PGPR, they reduce that inhibitory effect, that negative impact on growth. How these PGPR reduce the stress-induced negative impact? So the negative impact induced due to these stresses, that is reduced and you can see better growth. How that happens? That is through the production of plant growth regulators, through cytophore production, through induced systemic resistance, through enzymatic activity, through exopolysaccharides production, or through enhanced antioxidant systems. So these are the mechanisms how PGPR, they reduce the negative impact of these stresses on plant growth, and as a result, where you inoculated with PGPR, there is better growth, right? So if you have understood this picture, you can better understand the whole chapter. So how the abiotic stress tolerance takes place. So salinity and drought, the most common stresses, particularly in our country. These are the two most important stresses in our country, salinity and drought, the most common stresses. So these stresses cause, most of you are the soil science students, you know what salinity causes. So reduced diffusion, reduced nutrient uptake, restricted transpiration rate, less transport of nutrients from roots to shoots, impaired active transport, altered membrane permeability. So these are the effects of salinity or drought on plants. Increased sodium uptake and decreased potassium and calcium uptake. So every soil science student knows how these stresses, salinity and drought affects the plant, right? So these are the effects of salinity or drought on plant growth. And we have already discussed that whenever there is high level of, there is high stress, there is any stress, there is high level of ethylene production. Ethylene we discussed at a lower concentration. Ethylene is a plant hormone which improves the plant growth, and it is called as a ripening hormone as well, which helps the fruit in ripening. But when its concentration increases, that increased concentration, instead of promoting the plant growth, it inhibits the plant growth. High concentration of ethylene has inhibitory effect on plant growth. So what damages it can cause, it can have negative impact on roots. Particularly the root growth is affected and the roots remain small. Epinasty, plants become taller, the top tip starts growing up, premature senescence, abscion. These are the effects due to high ethylene concentration. So we need, whenever there is drought or a salinity stress, there will be more ethylene which will damage the plant in this way. So we have to reduce this high ethylene level if we want to avoid these negative effects. So how we can do that? One option is that we use plant growth promoting rhizobacteria, which is very easy and very economical approach. So plant growth promoting rhizobacteria is a potential tool for alleviating stresses. So whenever there is, how PGPR they can alleviate stress, that we will discuss now. But remember, I repeat this once again, that whenever there is any stress to the plant, that may be biotic stress, that may be abiotic stress. Whenever there is any stress to the plant, plant produces more ethylene, right? It is called a sort of stress hormone. So when more ethylene production, that inhibits particularly the root growth of plant. So if we want to avoid that harmful effect of stress, we have to reduce the high ethylene concentration. And one option is using PGPR. So how they do that? PGPR produce exopolysaccharides. And how these exopolysaccharides are helpful. These exopolysaccharides adsorb on clay particles and form a protective layer around soil aggregates and protect plant from desiccation. So, Umar, are you listening? Umar?
Yes, sir.
What is the difference between adsorption and absorption?
सर दोबारा 1 मिनट रिपीट कीजिएगा.
I have said that the exopolysaccharides absorb on clay particles. Why I didn't say that the exopolysaccharides are absorbed by clay particles? ADSORB. This word, look at this word. This is absorb.
सर एड्सॉर्ब का मतलब कि इनटेक करना सर
What is absorb?
सर इनटेक करना
If D की जगह पे, if instead of D it is B, absorb, then what is the difference? What is the difference between absorb and absorb?
सर एब्जॉर्ब का तो पता है कि सर मतलब इनटेक करना क्या कहते हैं अंदर की तरफ सर लेना मतलब और एड्सॉर्ब का सर के बाहर की तरफ मतलब सर टेक आउट
No.
आवाज शेर हो रही है
Try to tell what's the difference between absorb and adsorb. This is a very simple question, and you have to respond today. It's very simple. You have studied this in your lower classes, the difference between absorption and adsorption. No, Asanullah, wait. You have raised your hand, but just wait. I have to ask this from Umar.
सर नहीं सर सही तरह नहीं पता सर कॉन्सेप्ट नहीं क्लियर इसको
So you should try to understand the things. Okay, now two people have raised hand. Okay, Ruhullah,
यस सर
what is the difference between absorption and adsorption?
sir absorption sir between two similar things and absorption means between two dissimilar things sir
No. Ali Raza.
सर एड्सॉर्प्शन होती है अटैचमेंट ऑन द अपर साइड ऑफ क्ले पार्टिकल और एब्जॉर्प्शन होती है इनसाइड ऑफ क्ले पार्टिकल
Yes, beautiful, very good. Absorption means to take in, to uptake. For example, if I say this particular thing is absorbed by the plant, it means plant has taken it up in its body. But if I say it is absorbed on the plant, it means it is attached on the surface of the plant. It does not enter the plant, but it attaches on the plant. And you know what I said, that the exopolysaccharides absorb on clay particles. Clay particles living things तो नहीं है ना. These are not living things जो चीज़ uptake कर लेंगी. So the exopolysaccharides, they are just attached on the surface of the clay particles. Okay? If otherwise, I would have said that the exopolysaccharides are absorbed by plants, means the plants take in those exopolysaccharides. So here I am saying that the exopolysaccharides, Umar,
yes sir
did you understand the difference now?
yes sir
Okay, so now don't try to forget it.
okay
So exopolysaccharides, they attach on the surface of the clay particles, absorb on the clay particles, and form a protective layer around soil aggregates and protect plant from desiccation. So it's very clear. Had you just gone through this sentence, you would have understood the meaning of absorb. That the exopolysaccharides, you know, are the high molecular weight compounds, carbohydrates. They attach on the clay particles and they make a layer. And that layer surrounds the soil aggregates. And you know the soil aggregates have moisture inside. So when that layer surrounds the soil aggregates, as a result, the moisture from inside the soil aggregate does not come out, or it loses moisture at a lesser rate, right? So it is effective against drought conditions. So in this way, the plant is protected from desiccation. So if the soil has to dry, say under normal conditions, soil dries in 2 hours, but if there are bacteria which are producing exopolysaccharides, they will produce a layer around the roots and around the roots and the soil particles, so that soil will dry after 4 hours. So the water will remain available for more time. So in this way, the plant is protected from desiccation. Then these exopolysaccharides also are helpful in improving soil aggregation. So yeah, you studied this in your physical properties of soil, that the exopolysaccharides, when they form a layer around the soil particles, that is converted into an aggregate. So more the aggregation, more the moisture holding capacity of the soil, more the porosity of the soil. So they improve the soil aggregation. They help the PGPR for root colonization, right? So exopolysaccharides
Sir,
help the
ethylene
plant
जो
to colonize on the plant
ethylene
roots.
है
And I have
एक
already explained
एक कैसा
that,
जो
please mute your mics. So PGPR, if they can colonize the roots better, they can have better effects on the plant growth. So exopolysaccharides help the plant to have better colonization on the plant roots, and they help in the channelization of water and nutrients to plant roots. They help the plant to get more water and nutrients from plant roots. And they form biofilms. Biofilms are actually, you can say, the barriers which do not allow the moisture or the which do not allow particularly the pathogens to grow on the roots. This is a sort of layer. When a layer of microorganism attached with some surface, it is called as biofilm, right? So the microorganisms, they form a biofilm, and that biofilm does not allow the pathogens to attack the plant roots. Root adherence of soil. So exopolysaccharides help the roots to have better contact with the soil, right? And when they have better contact, they can better take up nutrients and the moisture from the soil. Then another mechanism is ACC deaminase activity. So I did say, I'll explain it later on. So ACC deaminase activity. So I repeat that ACC stands for 1-aminocyclopropane 1-carboxylic acid. 1-aminocyclopropane 1-carboxylic acid. This is Aswad Shahir. He is not trying to enter into the class. So it is not fair. I'm not going to allow him to enter into the class now after 1 hour. So ACC deaminase activity. PGPR, they secrete enzyme ACC deaminase that hydrolyzes ACC into ammonia and alpha ketobutyrate. I'll explain it in the next slide. The rhizobacteria bound to plant roots act as a sink for ACC, the immediate precursor of ethylene, and thereby lower the level of ethylene in a developing seedling or stressed plant. So as I said, I will explain all these in next slide from a picture. ACC deaminase activity containing PGPR inoculation improves plant growth, root, shoot length, yield, and nodulation under stress condition. So this is a model which has been proposed by a Canadian scientist, Glick. So this is ACC, 1-aminocyclopropane 1-carboxylic acid. Assume this is a plant seed or root. This side is a plant seed or root, and we have inoculated it with a PGPR. So a PGPR is attached to the seed or to the root. Now what happens? Whenever there is any stress, salinity stress or drought stress, what happens is that there is more production of ACC in the plant. There is actually, if the bacteria that producing IAA, IAA stands for IAA stands for indole acetic acid, which is a hormone. So that indole acetic acid enters the plant and it improves the cell elongation and root proliferation. So root growth is improved. Now, due to this IAA, there is more ACC synthase enzyme. Due to that stress, there is more production of ACC in the plant, and this ACC is converted into ethylene. So this is the reason why under stress condition, more ethylene is produced. That due to stress, plant produces more ACC synthase enzyme, it produces more ACC, and that ACC is converted into ethylene. And when there is high ethylene concentration, root elongation is stopped. You can see it is stopped, root elongation is inhibited, right? And when there is less root elongation, there will be less plant growth, right? So this is how high ethylene concentration or how stress affects the plant growth. Now, when you inoculate with PGPR, what happens is, whenever there is more ACC in the plant, and you know diffusion means the movement of a compound from higher concentration to lower concentration. When there is more ACC production inside the root, that ACC due to high concentration goes out of the root. And outside the root, there is a bacteria, PGPR, which has this enzyme ACC deaminase. So when this ACC goes out, this bacteria converts this ACC into ammonia and alpha ketobutyrate. So had this AC not come out, this would have been converted into ethylene. But now we have inoculated with a bacteria which can produce ACC deaminase. So this ACC comes out of the plant root, and here it is, instead of converting into ethylene, it is converted into ammonia and alpha ketobutyrate due to this enzyme ACC deaminase. And this ammonia and alpha ketobutyrate is used as nitrogen source by the PGPR. So in this way, when there is less ACC inside the plant, so there will be less ethylene production. So there will be less harmful effect of high ethylene concentration due to stress, right? So now if anybody could not understand this, he can ask me the question. I can stop here, but I have to continue the lecture. But I want you people to understand it perfectly, because this will help you much in your next classes. If anybody could not understand, please ask from anywhere.
Sir, you just start from the start. Adomet. What is the purpose of Adomet?
Sorry.
Sir, first step.
Okay. I
ADO
intentionally did not mention this whole pathway. This is adenosine methionine. This is the whole pathway, how ethylene is produced inside the plant. This is not whenever in the plant, it is not directly one compound is converted to the other compound, one compound is converted to the other then to the next, to the next, to the next. In this way cycle, like for example, plant takes up nitrogen, it is converted into one amino, into ammonium. Ammonium is converted into, say, one amino acid. That amino acid is converted into another amino acid. That is converted into protein. Similarly, first adenosine methionine is produced. That is converted, we can say it methionine. First methionine is produced, and that is then converted into ACC, one amino cyclopropene, one carboxylic acid, and that is then converted into ethylene. So we are, you can say, as A is converted into B, B is converted into C. It is like that. If you remove B, A will not be converted to C, right? So similarly, this methionine is converted into ACC, and ACC is converted into ethylene. So what happens with the inoculation is that this ACC, instead of going this way, due to concentration gradient, this goes out to the bacteria, where bacteria has produced this enzyme and converted this into ammonia and alpha ketobutyrate, right? And in this way, this inhibitory effect of ethylene is removed. Is it clear now?
Yes, sir. And sir, ACC is produced during stress condition.
Yes, more ACC is produced. ACC is produced under normal conditions as well, but under stress conditions, it is produced more.
Okay, sir.
And that's why that under stress condition, when there is more ACC production, so due to the concentration inside is more, so due to high concentration, it has to move towards lower concentration. So it moves out of the root where bacteria is present, and that bacteria is converting it. Yes, any other body, if he could not understand from anywhere.
सर इसमें इंडोलिक एसिड का जो है इसका रोल क्या है ये बता दें
किसका रोल?
सर ये डबल लिखा हुआ है.
Perhaps your speed is not good one. I could not understand you. Your voice is interrupted.
सर वो इंडोल एसिटिक एसिड का पूछ रही हैं कि उसका क्या रोल है.
Okay, इंडोल एसिटिक एसिड का क्या रोल है? इंडोल एसिटिक एसिड इज ए प्लांट हार्मोन. यहाँ पे एक रोल तो हमने यहाँ के पॉइंट ऑफ व्यू से सिंपली बता दिया है, सेल इलोंगेशन एंड प्रोलिफरेशन, राइट? सो यहाँ पे you should understand only that it is important for cell elongation. So it is actually a plant hormone, and you will study an independent chapter on plant growth regulators. There you will study its complete functions, right? And this IAA in this model, this also helps in producing ACC synthase enzyme. If more IAA is produced, this produces more ACC synthase. But its actual function as a plant growth regulator is cell elongation and root proliferation. Okay, so this is how ACC deaminase producing PGPR can help the plant in tolerating the stress. And this is actually why I have spent more time on this, is actually our group, soil microbiology and biochemistry at our campus. We are the pioneers of this work in Pakistan, using such bacteria for developing biofertilizers under stress conditions. So this mechanism, we are known not only in Pakistan, rather the world over, that we are working on ACC deaminase containing bacteria which have the ability of producing ACC deaminase, right? So I wish that you must know at least what is going on in your department, in your institute. So whatsoever the biofertilizers we have developed, those contain these typical characters that they have the ACC deaminase activity in them, in addition to some other mechanisms. Okay, we move forward. Then, so we have studied two mechanisms under stressful conditions. One was the exopolysaccharide production, second was the ACC deaminase, and third one is the antioxidants producing bacteria. So what are the antioxidant producing bacteria? So recall to your mind my last lecture where I mentioned the reactive oxygen species. Some people call them as ROS, abbreviate them as ROS, reactive oxygen species. These are present as singlet oxygen, hydrogen peroxide, and hydroxide ions. And these are developed in the photosystems of plants during stress. So whenever there is any stress, what can happen is the plant photosystem, you know that the plant has two photosystems, photosystem 1 and photosystem 2, and these photosystem 1 and photosystem 2 work together for photosynthesis. And whenever there is any stress, what happens is that these photosystems, they develop certain reactive oxygen species, which may be in the form of singlet oxygen, in the form of hydrogen peroxide, or in the form of hydroxide ions. And these, why these are harmful? Last time somebody asked the question, I replied briefly, but I said that I will discuss it in detail when we will go to that particular topic. So how these reactive oxygen species are harmful. So these denature the cell membrane, proteins, and DNA. So if there is more production of reactive oxygen species, then there will be more denaturation of cell membranes, proteins, and DNA. So some of the bacteria, the PGPR inoculation enhances the activity of antioxidant enzymes and reduces the negative impacts of stress. So antioxidant enzymes means they produce such enzymes which protect the plant from the harmful effects of those reactive oxygen species, right? So how they do this, the PGPR, what are those enzymes which protect the plant from the effect of reactive oxygen species? So they produce antioxidant enzymes. What are those antioxidant enzymes? These include superoxide dismutase, superoxide dismutase, peroxide dismutase, catalase, glutathione reductase. So these are the antioxidant enzymes. So if your plant or your bacteria is producing these enzymes, means there will be less harmful effect of those reactive oxygen species. So if you inoculate your plants with such bacteria which have the ability of producing these enzymes, what will be the benefit? The benefit is that the harmful effects of reactive oxygen species, which we discussed in the last slide, those are avoided, right? So then what are the osmoprotectants? The soil science people, they must know what are the osmoprotectants. Salman Niaz, Nasir Ahmed.
Sir, osmoprotection means sir, ये osmo
Okay, Rula is available. What is osmoprotectant? Okay, I don't want to waste time. If you don't want to respond, I tell you what are the osmoprotectants. They enhance production of osmoprotectants, and when you inoculate with PGPR, they enhance the production of osmoprotectants by PGPR. So what is the osmotic effect? Whenever there is more salinity, you are growing plant in a saline soil, what happens is, due to, more the salts, more negative the water potential. And water always moves from positive to negative water potential. So when there is saline soil, the water potential outside the plant will be less and inside the plant will be more. So what will happen is all the water from the plant will move out of the plant, and as a result, the plant will die, right? So to counter that, what the plant does, it produces some compounds inside its body, which like proline, glycine, betaine, these compounds are called as osmoprotectants, okay? Which similar is the case with PGPR also. Some PGPR, they can, if you try to grow those microorganisms under salt-affected conditions, so they can also suffer from the same issue that their water can come out. But they also produce certain osmoprotectants inside their body, and they then can survive in the salt-affected conditions. So there is enhanced production of osmoprotectants by PGPR. So if you have PGPR, there will be more production of osmoprotectants. And I told you proline, glycine, betaine, all these are the osmoprotectants. These help the plants to maintain internal water potential for better uptake of water and nutrients. I have just explained when there will be more production of osmoprotectants inside the plant, there will be not more positive water potential. Instead, the water potential inside the plant will also decrease. So the water from inside the plant will not come out. So the plant will survive under salt affected conditions also. So you people have studied this in your salinity classes.
Yes.
So this was how we can use the bacteria under stress and normal conditions. Now, the possibility is that you use a single type of bacteria or you use a combination of bacteria. These days, people are going towards using a combination of bacteria. And a combination of bacteria which can live together is called as bacterial consortium. If you remember when we were discussing the interactions, at that time also I mentioned this word consortium. So consortium means a combination of bacteria which can live together. So instead of having beneficial effect of only one bacteria, you have 2-3 types of bacteria which can live together and have separate effects. For example, one is solubilizing phosphorus, other is solubilizing potassium, other is producing antibiotics. So you combine all these 3 bacteria and prepare a product. So wherever you will apply that product, there will be more phosphorus solubilization, more potassium solubilization, and more disease control due to antibiotic production. So these days, people are moving towards developing biofertilizers using bacterial consortium instead of using a single strain inoculum. So what is bacterial consortium? There is difference in bacterial survival and effectiveness in a laboratory and field conditions. So say for example, you are using only one bacterium. You tried it in under lab conditions and it worked very well. It solubilized the phosphorus under lab conditions. But when you applied that phosphorus under field conditions to the plant, it did not survive over there. So it means your product is lost because it had only one type of bacteria. But if you have a consortium of bacterium and you apply that consortium to the field, then at least some of those type of bacteria will survive and will have at least some beneficial effects, right? If all can survive, well and good. If all cannot survive, at least some can survive and they can provide the beneficial effects. So there is always difference in bacterial survival and effectiveness in laboratory and field conditions. That might be due to low quality of the inocula. Whatsoever the inoculum or the product you are adding to the soil, it is of low quality. It does not have live bacteria in proper population, or it has inability of the bacteria to compete with the indigenous population. Already the bacteria which are present in the soil, they are more competitive. So the bacteria that you are adding to the soil, those are unable to compete with those indigenous population. So that's why your whatsoever the inoculum you have added, that is not effective. So under such conditions, then application of multi-strain PGPR is more efficient technique than applying a single strain inoculum. So that's why we try to use a combination of strains. So combination allows bacteria to interact synergistically. We have already studied what is synergistically, mutually beneficial for each other, but not obligatory. They provide nutrients to each other and also to the plant. They remove inhibitory products. They improve growth of each other. So what is the inoculant technology? So this is how we are discussing how the PGPR can be used. So by now we studied that you can use PGPR for improving plant growth, and I use the word inoculation, inoculant. So inoculation or inoculant means to add your desired population to the soil so that its population is increased. So this is called as inoculant technology. So what is inoculant technology? How you can formulate a microbial inoculant? Whatsoever the inoculants are available in the market, like we have some Rhizogold or Burak or others. Similarly, Nifty people have Biopower, like others. So different microbial inoculants are available. So how the formulation of microbial inoculants, you will cover this in your practical portion also. So I'll be a little bit quick because you will cover this in practical also. So how you can formulate the microbial inoculants. So PGPR strains have great potential to be formulated as biofertilizers, right? So inoculum means you have a higher population of a desired bacteria, which you have established in the lab that it is beneficial. Then you can apply that bacteria to the soil to have its beneficial effects on plant growth. This is called as inoculant. So commercial formulation of Bacillus species is used as biocontrol agents. So these days in the world, people are using commercial formulation of Bacillus bacteria for biocontrolling certain diseases. Then the formulation of Bacillus thuringiensis is used to control insect pests. So before this BT cotton, they were actually applying these Bacillus thuringiensis to control the insect pests. Then Pseudomonas putida or Panibacillus and Bacillus species, these are formulated to enhance the growth and yield of wheat. Like Rhizobium is applied to improve the yield of leguminous plants. So in microbial formulation, maintaining bacterium in metabolically and physiologically active state is an important aspect. So actually, the success of the microbial formulation on inoculation technology is that your formulation has metabolically and physiologically active bacteria in it. If those are dead, those are metabolically not active, then these are of no use. So you have to have a product which is metabolically active, which is physiologically active. So in certain environmental conditions where single strain inoculum is unable to perform better, the development of multi-strain inoculum can be very effective. So I have already explained that how a single strain cannot be that effective as is a combination of multi-strains. So there may be different options of preparing that formulation. For preparing that formulation, you can have some carrier material. You grow those bacteria and you can add that bacteria into some carrier. Like, for example, you take any pill, any tablet, say Panadol. The actual medicine is very little one in it. More is the, you can say, the carrier material. ठीक है यार पर यार अल्फाफा लेके आओ की होया जे तुम So carrier material plays important role in formulating microbial inoculants. So for example, this is I'm showing you a product from this lab. You can see this is a carrier based product. which has 3 types of bacteria, means a consortium of bacteria is present in it, which can be used for controlling the weeds. This is actually a bioherbicide, right? So this is the inside is some organic material and the bacteria. So that organic material is called as actually the carrier material, right? So we grow the bacteria in lab, and then we harvest those bacteria and add a very high population of those bacteria into that bag, and these bacteria continue growing over there. These act as this carrier material, actually it acts as delivery vehicle to transfer live microorganisms from laboratory to the rhizosphere. So what you do is, we generally recommend that you open this bag and you apply it to the seed and then sow the seed in the field. And when the seed will germinate, it will have bacteria on the surface of the seed. So those bacteria will stick to the plant roots, and in this way, this can provide the beneficial effects. So this is the carrier material. So carrier material also provides a suitable amount of PGPR with good survival efficiency. So we have to select such carrier material where the bacteria can survive for longer time. If it cannot survive for longer time, then of course it is of no use. It provides a suitable microenvironment for the PGPR